Assalamu alaikum. Hello learners. You are welcome to my class. So let's start. Here is a male person, is a female person. Here is another male, another female. This couple has a son and this couple has a daughter. The son and this daughter of these two different families get married, so they have a baby. The son carries the characteristics or traits of both his father and mother. This daughter carries the characteristics or traits of both her father and mother. This baby carries the characteristics both its father and mother. So here we can see that the passing of traits or characteristics occur from one generation to another generation. Now, what is called the passing of traits from one generation to another generation? The passing of traits from one generation to another generation is called heredity. So we can say that heredity is the passing of traits from one generation to another generation. You have already learned that passing of traits occurs from one generation to another generation, and it is called heredity. Heredity occurs through some materials or substance which are known as hereditary material. Chromosome, DNA, RNA, gene act as hereditary materials. Here is a cell. It's a eukaryotic cell. It is nucleus. It is chromosome, and it's a DNA molecule. Nucleus remains inside the cell. Chromosome remains inside the nucleus, and chromosome consists of DNA molecule. Dear learners, you know chromosome remains inside the nucleus. It's a chromosome, and this coiled structure is chromatin. This is nucleosome. Nucleosome contains 8 histone protein. This round structure is histone protein, and this histone protein are coiled by two coiled by a thread-like structure, and this thread-like structure is DNA. Chromosome consists of highest condensed structure of the DNA double helix. So from this figure you can say this chromosome consists of the highest condensed structure of DNA double helix. This is double one helix and another helix. That means double stranded structure. DNA is a double stranded structure. And chromatin consists of the unraveled condensed structure of DNA for the purpose of packaging into the nucleus. When chromosome remains inside the nucleus, then this structure is called chromatin. What is the difference between chromosome and chromatin? In chromosome, you can find the highest condensed double helical DNA structure. In chromosome, you will get, mind it, highest condensed structure of the DNA double helix, but in chromatin, you will get unrevealed condensed structure of DNA. And why this DNA remain unrevealed in chromatin? Because this chromatin has to have inside the nucleus, and this one is DNA. Nucleosome is composed of a little less than 2 turns of DNA unwrapped around a set of 8 proteins called histone. These round-like structures are known as histone protein, and here you can see 2 turns of this thread, and you have already learned that this magnified structure of thread and it is DNA. So nucleosome consists of 2 turns of DNA and 8 histone protein. Now we will try to learn about the structure of a chromosome. It's a chromosome. In its structure, we can find 2 chromatids, telomere, its short arm is known as P arm and long arm is known as Q arm. These 2 chromatids get attached at the centromere. Here it is centromere, and we know chromosome consists of DNA molecule. It's a DNA molecule. A chromosome consists of a DNA molecule, but here you can see 2 DNA molecules. Why? This chromosome is at the time of cell division. This chromosome indicates the chromosome at the time of cell division. That's why here DNA replication is occurred. So you can see 2 DNA molecules in here, but you will get 1 DNA molecule in 1 chromosome. Chromosome is a thread-like structure in which DNA is tightly packaged within the nucleus. You have already learned that each chromosome is made up of a single molecule of DNA tightly coiled many times around proteins called histone that support its structure. So inside the chromosome you will get DNA and histone protein. The structural unit of a eukaryotic chromosome is the nucleosome. Now can you tell me what is nucleosome? Just remind it. In a nucleosome there are 8 histone proteins and 2 turns of DNA. The most important function of chromosomes is to carry the basic genetic materials DNA from parents to progeny. And dear students, you can see the DNA molecule inside the chromosome, and you also know DNA carries the traits from parents to progeny. That means parents to son or daughter, that means next generation. Dear learners, you have already known that chromosome consists of DNA. Now we will learn the structure of DNA. It's a DNA, but not a DNA molecule. It's a part of DNA molecule. DNA is usually a double-stranded spiral structure of polynucleotides. A strand is complementary to other. It is a double-stranded structure. It is like a twisted ladder. It is one strand and it is another strand. And mind it, it is a spiral structure of polynucleotides. A human DNA is about 1.8 meters, that's 5 feet long. 5 feet long, 1.8 meters or 5 feet long. So you can consider DNA as a large molecule. In it, there are 5 carbon sugars, deoxyribose sugar, nitrogen bases, adenine, guanine, cytosine, thymine, and inorganic phosphates. These 3 components collectively are called nucleotide. And you know, DNA consists of polynucleotides. So in a nucleotides you will get deoxyribose sugar, nitrogen base, and inorganic phosphate. Deoxyribose sugar, nitrogen bases, and inorganic phosphate combinedly form nucleotides. And DNA is formed with many nucleotides. That's why it is called a structure of polynucleotides. Nitrogen bases are of 2 types, such as purines and pyrimidine. Adenine and guanine are purines. Cytosine and thymine are pyrimidines. Mind it, nitrogen bases are of 2 types. In DNA you will get 2 types of nitrogen bases. One is purines and another is pyrimidine. Adenine, guanine are purines and cytosine and thymine are pyrimidines. Adenine of one strand bonds with a thymine of another strand by 2 hydrogen bonds. And guanine of one strand bonds with cytosine of another strand by 3 hydrogen bonds. So we can say a bond is always developed between purine and pyrimidine. Adenine is bonded with thymine. Adenine is purine, thymine is pyrimidine. Again, guanine is bonded with cytosine. Guanine is purine and cytosine is pyrimidine. So bond is always developed between purines and pyrimidines. Here is purine, pyrimidine. If you consider it is pyrimidine, it will be purine. If it is pyrimidine, it will be purine. And these strands are composed of 5 carbon sugar and inorganic phosphate. And you can consider the handle or helix of a twisted ladder, this spiral structure, and the step of the ladder will be nitrogen base. These are the steps of the ladder. If you consider this structure as a twisted ladder, the handle of the ladder will be these 2 sides and the step will be the nitrogen base. A complete twist in a helix is 34 angstrom. Angstrom is small unit. 1 angstrom equal 10 to the power minus 10 meter long, and in a complete twist there are 10 nucleotide pairs. So the length between two adjacent nucleotides is 3.4 angstrom. It is a complete twist, and in this complete twist you will get 10 nucleotides. Now what is nucleotides? You know, nucleotides composed of deoxyribose sugar and a nitrogen base and an inorganic phosphate. Deoxyribose sugar, nitrogen base and an inorganic phosphate collectively called nucleotide. You will get 10 pairs of nucleotide in a complete twist or complete turn of a DNA structure. You will get clear idea about the structure of DNA from here. It's a molecular structure of DNA. You know, DNA is composed of deoxyribose sugar. It's a 5 carbon sugar. Phosphate, inorganic phosphate. Deoxyribose sugar is indicated by S. Inorganic phosphate is indicated by P. Adenine, guanine, cytosine and thymine. Adenine, guanine, cytosine and thymine are collectively called nitrogen base. Nitrogen base. DNA is composed of deoxyribose sugar, phosphate and nitrogen base. Adenine and guanine are called purines. Cytosine and thymine are called pyrimidines. Nitrogen bases are of two types, purines and pyrimidines. It's a deoxyribose sugar and a nitrogen base. It may be adenine, it may be thymine, maybe guanine, maybe cytosine. It is called nucleoside. A deoxyribose sugar and a nitrogen base collectively called nucleoside. It's a deoxyribose sugar, inorganic phosphate, a nitrogen base. It may be adenine, it may be thymine, it may be guanine, it may be cytosine. It's collectively called nucleotide. If one inorganic phosphate gets attached to a nucleoside, it will form nucleotide. Nucleoside, nucleotide. You know, DNA is a structure of polynucleotide. DNA contains huge number of nucleotide. It's a nucleotide with adenine nitrogen base. It's purine nitrogen base, purine. It's another nucleotide having pyrimidine nitrogen base, thymine. Adenine nitrogen base, thymine. Purine nitrogen base and pyrimidine nitrogen base bonded with double hydrogen bond, hydrogen double bond between adenine and thymine. Another nucleotide having nitrogen base guanine, that means purine. It's a triple hydrogen bond. Another nucleotide having cytosine. It's pyrimidine nitrogen base. So bond is always formed between a purine nitrogen base and a pyrimidine nitrogen base. If the purine nitrogen base is adenine, the pyrimidine nitrogen base will be thymine. But if the purine nitrogen base is guanine, the pyrimidine nitrogen base will be cytosine. Between adenine and thymine there will be hydrogen double bond, and between guanine and cytosine there will be hydrogen triple bond. Another nucleotide having Thymine. Thymine is what types of nitrogen base? It is pyrimidine. So this side there will be purine nitrogen base. Adenine is purine nitrogen base. Mind it. Pyrimidine, purine. Here is purine, pyrimidine. Purine, pyrimidine. But here is pyrimidine, purine. It's purine and pyrimidine. Adenine, purine, thymine, pyrimidine. It's a pyrimidine. C is pyrimidine and guanine G is purine. So bond always formed between purine and pyrimidine or pyrimidine or purine. It's a molecular structure of DNA. Molecular structure of DNA. It's a double bond, triple bond. Double bond between adenine and thymine or thymine or adenine, and triple bond always in between guanine and cytosine or cytosine and guanine. I think you have got clear idea about the molecular structure of DNA. Now we learn the structure of RNA. RNA is ribonucleic acid, DNA is deoxyribonucleic acid, RNA is ribonucleic acid. You know, nucleic acids are of two types. One is DNA and another is RNA. This structure indicates RNA. You can see the double-stranded structure or double strand in DNA, but here you can see single strand in RNA. Here also have nitrogen base. You can see 4 nitrogen bases are here: adenine, guanine, cytosine, uracil. There is no thymine. Instead of thymine, there is uracil. It's a structure of RNA. Single structure, single stranded structure. RNA stands for ribonucleic acid. Most of the RNAs are single stranded. It is composed of 5 carbon ribose sugar, inorganic phosphate, nitrogen base adenine, guanine, cytosine and uracil. Instead of thymine, uracil is present here. In DNA, you have got deoxyribose sugar, but in RNA, you have got ribose sugar. Many types of RNA. The 3 most well known are messenger RNA, transfer RNA, ribosomal RNA. RNA is also genetic material or heredity material where DNA is absent. RNA acts as hereditary material where DNA is absent. You will get RNA as genetic material in some viruses, RNA viruses. Suppose tobacco mosaic virus, it's a RNA virus. There is no DNA in this virus. Here RNA acts as genetic material. Dear students, you have learned hereditary materials. The materials that passes traits from one generation to another generation are called hereditary materials. You have learned 3 hereditary materials: chromosome, DNA and RNA. That's all about our today's class. Thank you. Assalamu alaikum.